media that are designed with specific growth factor to support previously unculturable microorganisms. Such examples of microorganisms are microorganisms that grow in extreme regions. Now we can culture them in a much easier in isolation before strict plating and dilution technique were the primary method for pure isolation. Physical separation techniques also were used, like centrifugation, like filtration, but they were often limited in efficiency. Identification also heavily relied on gram staining and biochemical reaction. But now, we now have things like the use of fluorescent activated cell sorting FACS, which use fluorescent markers to separate microbial populations based on specific traits. Another thing that is also used now sorting called marks. They use magnetic bead to isolate target microbes for population. So the bead are coated with specific genes that are expected from the microorganism. So the microorganism, when the medium is passed through, is able to attach to the bead that is being run through. Also, method helps us to before. Identification relied heavily on gram staining, colony, morphology, biological tests, strips, and limited biological assays. And it was slow and labor intensive, with results taking up to days or weeks. And it could also be prone to misidentification due to phenotypic similarities among these species. But now we have 16S RNA sequencing, which rapidly identifies microorganisms by analyzing conserved ribosomal gene sequences. We also have whole genome sequencing. It provides highly detailed microbial identification and insight into antibiotic resistance and virulence factor. Another machine, which is in this figure 7, is the image of Malditov. It is called metric assisted laser desorption ionization time of flight mass spectrometry. It is used to quickly identify microbes by analyzing their protein profiles. Another mechanism used now is the next generation sequencing and metagenomics. It identifies entire microbial communities without culturing. Another method used now is the PCR, polymerase chain reaction, and quantitative PCR. It enables fast and precise microbial detection, including pathogen identification in clinical environmental samples. Then AI and machine learning in identification. Automated analysis of sequencing and imaging data proves accuracy, and it also reduces human error. That is, when we use machine, it also makes our work faster and reduces human error. So here is an image of the Malditov machinery. So what are the limitations to this advanced method? The limitation to this advanced method is that techniques like Malditov MS, they are powerful and their accuracy depends on comprehensive databases. And misidentification can occur if a microbe is not well represented in the database. So since it is based on data collection, if there is not enough data, that is it leads to misidentification and error. So that means that concerning advanced method like this, we still need to have additional databases that will help us to make our work error-free. Then another important fact, which is also a limitation to this advanced method, is cost consideration. advanced molecular technique like next generation sequencing can be expensive and not readily accessible to all laboratories. We can also say as a proof that it's not every laboratory that has all this machinery due to costs of some of this machinery, and sometimes could also even be due to the fact that not trained on the use of this machinery. So these are the limitations to this advanced method. In conclusion, advancements in microbial culture as isolation and education have made the process faster. It has made it more efficient and more comprehensive. The shift towards molecular and high throughput techniques allow for the identification of or unculturable microbes, improving our understanding of microbiomes in health, industry, and the environment. These are my references. Thank you for listening.
Thank you very much. Advances in microbial culturing, isolation, and identification. Do you have any question for her? Any question for her? Okay, Ajadi, you want to ask her a question?
Thank you. Yes, sir.
Go on, sir. Go on. Ask your question. Ajadi, ask your question.
Can you do this in identification of bacteria? Miss Ifeoluwa.
Yes, sir.
Please, can you? Hello.
I heard your question.
Okay.
What this method does is to amplify small fragments of the cysteine and sequences the fragments and compare the sequence to reference sequences in database.
Are you satisfied with the response, with the answer? Any other question, please?
I'm okay, sir.
Any other question? All right. If there's no other question, thank you.